Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 1B for May 2023. Let us begin with the first question. Question 1. All living organisms share characteristics. State two characteristics that all living organisms share. Living organisms can respire, they carry out nutrition, excretion, reproduction, they respond to stimuli, they carry out homeostasis, they grow as well as moving. The next part says, some organisms are pathogens. Which of these organisms can cause a bacterial disease in humans? This is pneumococcus. And in part B, viruses are pathogens but not living organisms. Describe the effect of a named virus that infects plants. Tobacco mosaic virus infects plants. And an example of the symptoms is it causes yellow discoloration in the leaves, and this is what we can see here. And here they say, give three differences between the structure of viruses and bacteria. Viruses have a protein coat, but bacteria don't. Bacteria have flagella, but viruses don't. And bacteria have cytoplasm, but viruses don't. So this brings us to the end of question 1. Let's continue to question 2. The diagram shows part of a food web from an ecosystem. So we can see the food web there. They say using the information in the food web, draw a food chain that contains 5 trophic levels and include the birds. Birds are over here. If plants are the producers, we can see this is the first food chain going all the way towards the birds. It includes the plants, the worms that eat roots, the small arthropods, large arthropods, as well as birds. There is also another food chain including plants, fungi, again small arthropods, large arthropods, and then birds. So those are the ones I drew here. When you draw a food chain, the arrow has to face towards the organism that is the consumer. So in this case, we can see the fungi are eating the plants and the small arthropods are eating the fungi and so on. And here they say describe how bacteria and fungi obtain energy from the organic matter. They do so by decomposition of substances. They release enzymes or secrete enzymes onto the food sample. External digestion takes place, and then the nutrients are going to be absorbed in order for respiration to be carried out to produce ATP. In part B, a pesticide gets into this ecosystem. The pesticide kills small and large arthropods. Explain how this affects the population of birds and the population of worms that eat roots. If I take you back here, if the pesticide affects small arthropods as well as the large arthropods, the birds will have less food to eat. Actually, we can see this bird is only eating these large arthropods, so they will not have food and their population is going to decrease. So here I say since birds feed on large arthropods, destruction of the arthropods would lead to fewer birds. The worms will also increase in number since they are only eaten by small arthropods. So the population of the prey of the small arthropods, which is going to be the worms, is going to increase in number as well. In part C, scientists use 3 different traps to collect some soil animals. These traps are a basket trap, a pitfall trap, and a cul-de-sac trap. The number of animals collected by each trap are counted each day. The animals collected are classified into 3 types. They are springtails, they are mites, and large arthropods. So they say the graph shows the scientists' results. On the vertical axis, we have the mean number of animals collected each day, and on the horizontal axis, we have the different animals collected. We can see the key is given here. We have the basket trap, the pitfall trap, as well as the cul-de-sac trap. Looking at all the organisms, we can see mites were the most collected, and the pitfall trap collected the least, while the cul-de-sac as well as the basket trap collected almost equal numbers. With all organisms, the pitfall trap collected the least number of organisms, and the most collected organisms were the mites, while the least collected were the large arthropods. So here they say discuss the number of each animal type collected by the 3 traps. They want you to refer to the scientists' results in your answer. Since I've already explained the results in the table, let me just go through what I wrote. I say the greatest number collected was for mites, and the least was for large arthropods. Cul-de-sac traps collected the most number of animals, while pitfall traps collected the least. Cul-de-sac traps collected most mites and large arthropods. The basket traps collected the most springtails. The soil contained more mites, and the number of large arthropods was the least. The number of mites collected by the basket traps and cul-de-sac traps is almost the same. And again, you could write more based on what you see from the table. So this brings us to the end of question 2. Let's continue to question 3. In question 3, the diagram shows a plant cell with some structures labeled. So we can see the cell. And they ask which structure is the cell vacuole. Structure T is the vacuole. So D is my answer. And the next part says which structure is the site of photosynthesis. Photosynthesis takes place in the chloroplast. And we can see structure P is the chloroplast. This one here is the mitochondrion. So P is going to be the answer there, and which is going to be A. The next they say which structure is the cell wall. If I take you back here, the cell wall is going to be structure S. So my answer here is going to be a C. And next they say structure U is the site of protein synthesis in the cell. What is the name of structure U? Protein synthesis takes place at the ribosome or in the ribosome, and therefore we can say C is the answer. The next part says a cell is shaped like a cube. Each side has a length of 0.05 millimeters. Calculate the surface area to volume ratio of this cell and give your answer in the form of n/1. So if it's in the form of a cube, surface area of a cube is 6 times the length squared, which is 6 times the length given here. When you square that and multiply, my answer was 0.016854 millimeters squared. And again, because it's a cube, the volume is equal to length cubed, which is 0.053 power 3, giving me 1.4888 times 10 power negative 4 millimeters cubed. So the surface area to volume ratio is going to be that to that here, and I got 113 to 1. So that was my answer here. In part C, animal cells, unlike plant cells, do not have a cell wall. Explain how this difference affects red blood cells when placed in distilled water. When cells have no cell wall, it means they can take up water excessively and they can burst. My answer was they take up water by osmosis from a region of high water potential to a low water potential region. And when they do that, they will swell, and due to lack of a cell wall, they can take up water and swell excessively until they burst. And down here they say, explain how this difference affects red blood cells when placed in a concentrated salt solution. And again, when placed in a concentrated salt solution, they will lose water excessively by osmosis from a higher water potential, which is inside the cell, to a lower water potential, which is outside. And if they continuously lose water, they will become shrunken or shriveled. So this brings us to the end of question 3. Let's continue to question 4. In question 4, the diagram shows a human reflex response to touching a hot object. Some of the structures in the reflex arch are labeled. So we have the hot object here, we have the spinal cord and the different structures which are labeled. And they say give the name of this reflex response. The hand is going to be withdrawn immediately, so it's a withdrawal reflex, which is going to be involuntary. And they say draw an arrow on the diagram to show the direction of the nerve impulse at point X. Point X is at the motor neuron, so the nerve impulse is coming towards the muscle, so my arrow was in this direction. The next part says which structure detects the stimulus. The stimulus is going to be detected by the receptor. In this case, it's going to be in structure E. This is where the receptor cells are going to be. So my answer was A. And down they ask which structure is the motor neuron. The motor neuron is going to be structure I. So my answer is a C. And in part B, a neuron is 1.10 meters in length. The speed of the nerve impulse in this neuron is 120 meters per second. Calculate the time in seconds for the impulse to pass along the neuron and give your answer in standard form. We know time is equal to distance over speed, so my distance is 1.1 divided by the speed, and I get a time of 0.0092 seconds. This should be S. Let me take away the E. And then they say describe how the impulse passes from neuron to neuron in the reflex arc. Between two neurons there is a synapse, and one neuron where the impulse is coming from secretes a neurotransmitter that diffuses through the synapse, and then attaches onto the next neuron for the impulse to be propagated in that next neuron. So I said between two neurons there is a synapse. At the synapse, a neurotransmitter is released from the neuron with impulse. The neurotransmitter diffuses through the synapse and it binds onto the next neuron to stimulate an impulse. In part C, pain has a survival function. If a person damages their ankle, they sense pain. Explain how sensing pain benefits an organism. Pain alerts somebody that they are hurt so that they can be more careful or take precaution. It also prevents them from further injury. It allows them time to rest so that they can heal, and then the organisms will be more careful in order not to be hurt again in that similar situation. Some medicines are used to reduce pain. These medicines work by preventing communication between the injured ankle and the brain. Explain which component of the nervous system may be affected by these medicines. Any part of the nervous system that prevents transfer of the impulse from the receptor to the central nervous system can prevent feeling pain. So it could be at the receptor, it could be through the sensory neuron, it could be at the synapse, and so on. So I say this could prevent receptor cells from converting the pain stimulus into electrical impulses, or the medication could prevent transmission of impulses in the sensory neuron and the relay neurons. It could also prevent transmission of the impulse at the synapse. So this brings us to the end of question 4. Let's continue to question 5. In question 5, red blood cells and white blood cells are two of the components found in the human blood. State two other components of blood. Other components include plasma as well as platelets. And in part B, some white blood cells destroy pathogens. The diagram shows 4 stages in this process. So they want you to describe what is happening in each stage of this process. We can see in this stage, the pathogen is being engulfed, which is the bacterium being engulfed by the white blood cell. Here it's inside and the vesicle has formed around it. We call that a phagosome. And in the next step, this cell releases lytic enzymes. We can see the vesicles containing the lytic enzymes are fusing with the phagosome, releasing the enzymes that are going to digest the bacterium. And then after digestion, the products are going to be secreted out or released. So in stage 1, the white blood cells extend out the pseudopodia to engulf the bacterium. In stage 2, the bacterium is inside the phagocyte. A vesicle, in this case I'm talking about a phagosome as the vesicle and the bacteria trapped inside. This is formed around it. And in stage 3, the vesicle containing digestive enzymes fuses with the phagosome containing the bacterium. And in stage 4, digestion of the bacterium has occurred and the products of digestion are released by the phagocyte to the outside. In part C, the picture shows a human red blood cell. We can see this is the red blood cell. They say the diameter of this human red blood cell is 8.1 micrometer. Determine the magnification of this picture. We are given that 1 millimeter is 1,000 micrometer. The diameter of the image was 57 millimeters. So 57 millimeters is going to be 57,000 micrometer, or 57 times 10 power 3. Since magnification is equal to image size divided by the actual size, this is my image size and the actual size is given here. So 57 times 10 power 3 divided by 8.1 gave me 7037, and that was my magnification. Next they say, patients with reduced number of red blood cells are described as anemic. In an investigation, scientists measure the percentage oxygen saturation of blood and the rate of tissue deoxygenation. The rate of tissue deoxygenation is a measure of how fast oxygen is lost from the tissues. They use 4 groups of patients. Number 1, the non-anemic patients with normal blood flow. Then we have the anemic patients with normal blood flow, the non-anemic patients with slow blood flow, and the anemic patients with slow blood flow. The table shows the scientists' results. So we see the normal blood flow, non-anemic as well as anemic, and the slow blood flow, non-anemic as well as anemic. The corresponding results including number of patients, the mean percentage of oxygen saturation, we can see it's higher here in comparison to that and higher here in comparison to that. So we see the non-anemic have a higher mean percentage of oxygen saturation in blood. We also see the mean rate of tissue deoxygenation in percentage per minute, we see it's higher here and lowest here. So the normal blood flow that are anemic have a higher deoxygenation percentage, and those that are non-anemic in slow blood flow have a lower value, or they have the least value. So they say discuss the relationships between anemia, blood flow, percentage oxygen saturation of blood, and the rate of tissue deoxygenation. You should refer to the data in the table and use your biological knowledge in your answer. I've already talked about the results in the table, so I'm just going to go through what I wrote here. I said the percentage of oxygen saturation was higher in non-anemic than in anemic for both patient groups. The non-anemic patients in the normal blood flow group had the highest mean percentage of oxygen saturation. The anemic patients in the slow blood flow group had the least percentage of oxygen saturation. Anemic patients in the normal flow blood group had the highest rate of tissue deoxygenation in percentage per minute. And my reason, this is from my knowledge, anemic people have fewer red blood cells, so they have less hemoglobin, and therefore they carry less oxygen, leading to lower percentage of oxygen saturation in their blood and higher rates of tissue deoxygenation. The normal flow non-anemic and slow flow anemic had the same mean rate of tissue deoxygenation in percentage per minute. The differences in the results could also be caused by other factors like age, gender, and lifestyle. And again here, people who are younger and older have different metabolic requirements in the body, so they could have different percentage saturation of oxygen, or they could require different amounts of oxygen, so that can lead to differences. They didn't tell us their gender, their age, and lifestyle. We might not be able to make fair conclusions about the result we see. So this brings us to the end of question 5. Let's continue to question 6. In question 6, many characteristics of pea plants are genetically controlled. One of these characteristics is flower position. Flower position can either be axial or terminal. They say, in a first cross, scientists cross pea plants with axial flowers with pea plants with terminal flowers. The first cross produced 1,120 offspring plants. All of these offspring plants had axial flowers. So because all these plants had axial flowers, we can conclude that being axial is dominant. And because all of them had axial, it means they were all heterozygous, meaning the offsprings of the first generation were all heterozygous. So that tells us both parents were homozygous, one homozygous dominant and the other homozygous recessive. And again, the dominant one was the axial plant. Use a genetic diagram to show the genotypes of the parent plants, the gametes they produce, and the genotypes of the offspring plants. If I let capital A to be for axial and small a to be for terminal, since we've already established that the axial is going to be dominant, the parental genotypes are going to be capital A, and small a, and the gametes produced are going to be capital A and small a. If we do a genetic cross, the offspring genotype is going to be capital A and small a, meaning they're all going to be heterozygous, and therefore they will all be axial. That is going to be their phenotype. In part B, the scientists allow the offspring from the first cross to self-fertilize. The second cross produced 858 second-generation plants. 608 of the plants had axial flowers, and the other plants had terminal flowers. They wanted to calculate the ratio of the plants with axial flowers to the plants with terminal flowers, and they wanted to give your answer in n/1. The axial flowers are 608, and that means the terminal are going to be the total, which is 858 minus 608, giving us 250 flowers. So the ratio of axial to terminal is going to be 608 to 250, which is 2.43 to 1. And that was my answer here. Next, they say explain why the ratio of plants with axial flowers to the plants with terminal flowers is different from the expected ratio. If I take you back here, we can see these were all self-pollinated, and since they were all heterozygous, when heterozygous go through self-pollination, the ratio of the offsprings is going to be 3:1. So the expected phenotypic ratio is 3:1, but the observed phenotypic ratio is 2.43:1. It means there could be some differences. Maybe some organisms did not grow to mature, maybe because fertilization is a random process we cannot control, and so on. So I said the parents in the second genetic cross were all heterozygous, so the expected offspring phenotype ratio is 3:1. However, it was 2.43:1. This could be because fertilization is random or is a random process, so we can't control the outcome. It could also be that the offsprings did not mature or some offsprings did not survive, so their phenotype was not accounted for. And next they say scientists want to discover if plants with axial flowers produce more seeds than plants with terminal flowers. They want you to design an investigation to discover whether plants with axial flowers produce more seeds than plants with terminal flowers. They want you to include experimental details in your answer and write in full sentences. So here we need to set up experiments involving both axial and terminal flowers. We have to make sure whatever we use or the plants we use have to be the same species, they have to be the same age. And we will repeat the experiment using many axial and terminal plants under similar conditions in order to get the mean results. We will count the number of seeds produced by each plant, or you could measure the mass. Allow the plants to grow for the same period of time for seeds to be collected. During this experiment, we'll keep variables like temperature, carbon dioxide concentration, sunlight intensity, wavelength of light constant. And also because these plants are growing in the soil, we have to make sure that soil conditions are maintained in all experiments, so they have to be exposed to the same amount of water, same minerals, so that the only variable is the independent variable, which is the different types of flowers. So this brings us to the end of question 6. Let's continue to question 7. In question 7, lipase digests lipid into fatty acids and glycerol. The fatty acids change pH of the solution. A student uses this method to investigate the effect of temperature on the activity of lipase. In step 1, label a test tube with a temperature of 20 degrees and then add 5 drops of phenolphthalein indicator to the test tube. Then add 5 centimeters cubed of milk to the test tube. Then add 7 centimeters cubed of sodium carbonate solution to the test tube. The contents of the test tube will now be pink. This is because the sodium carbonate solution is going to be alkaline and the phenolphthalein indicator will turn into pink. Then place the thermometer in a test tube. Place the test tube in a water bath at 20 degrees Celsius for 5 minutes. Here the thermometer is going to measure the temperature of the water. Then place a beaker containing lipase in the same water bath for 5 minutes. This is to ensure that everything is at the same temperature before they're mixed. Replace the thermometer with a glass rod and then measure out 1 centimeter cubed of lipase from the beaker into the water bath. Add the lipase to the test tube, stir the contents and record the time until the contents lose their pink color. So this is the setup for the experiment. They say the student repeats this method for 5 more temperatures. There is 25, 30, 35, 40, and 45. In part A, state why the student leaves the test tube and the beaker in the water bath for 5 minutes in steps 6 and 7. This is done to ensure that the enzyme lipase and the substrate, which is the milk containing the lipid, are at the same temperature before mixing. And then in part B, they say give the dependent variable in this investigation. The dependent variable is the result. In this case, it's the time taken to lose the pink color. And next they say state one variable the student controls in their investigation. They need to control the volume of milk, the drops of phenolphthalein, the volume and concentration of lipase, and so on. And in part C, suggest the purpose of an phenolphthalein indicator in the investigation. It is there to show the endpoint to ensure that all lipid in the milk has been digested. In part D, they say the table shows the student's results. These are the results. And then they say plot a line graph to show the effect of temperature on the time taken for the contents of the test tube to lose the pink color. Use a ruler to join your points with straight lines. On the vertical axis, I have time taken for the contents to lose the pink color in seconds. And on the horizontal axis, I have temperature. Initially, I had studied this at 0, but my graph was so squeezed in a smaller space, so I expanded or I made the scale wider so that it can be visualized properly. So I chose a scale from 225 all the way to 400 on the vertical axis, and here we can see 20, 30, and 40 You have to position your points appropriately, draw the straight lines as required, and then you'll be able to get the 5 marks that are awarded here. In Part E, explain why increasing temperature affects the time taken for the contents of the test tube to lose the pink color. Increasing temperature will decrease the time taken to lose the pink color because the higher the temperature, more kinetic energy is going to be provided and the collisions are going to be faster for the rate to be faster. But this will occur only until the optimum temperature, and after that, the rate is going to slow down and it will take longer. So I said increasing temperature until the optimum temperature increases the kinetic energy, so more collisions occur between the enzyme and the substrate, and the reaction proceeds faster, and the time taken to lose the pink color will decrease. However, after the optimum temperature, increasing temperature breaks the bonds in the active sites, so the rate of formation of the enzyme-substrate complex will fall. Since the substrate will no longer fit into the active sites, the rate is going to slow down, and here I could write, so it will take longer to lose the pink color. I've just added this statement, until the time when it will no longer lose the pink color. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Yeast can be used in experiments to investigate the effect of different concentrations of glucose solution on the rate of anaerobic respiration. The diagram shows apparatus used to measure the rate of anaerobic respiration in yeast. So we can see there is a water bath to maintain the temperature, there is a thermometer to check the temperature, there is a suspension. The oil layer is there to prevent any oxygen from entering since this is anaerobic respiration. And then we have liquid A. They say give the function of the water bath. This is to keep the temperature constant. And then in part B they say liquid A can be used to identify the gas released during anaerobic respiration by the yeast. Explain which substance can be used as liquid A. Here you could use hydrogen carbonate indicator, and this is going to turn yellow. Or you could use lime water, which will turn cloudy if CO2 is present. And in part C, a student wants to accurately measure the rate of anaerobic respiration in yeast using this apparatus. State what additional apparatus they would require. Because the aim is to measure the rate, it means we need a change in something divided by time, so we will need a stopwatch to check the time. However, because CO2 is produced, we will need a way to collect the volume, so a syringe is going to be necessary to collect the volume of gas produced so that we can calculate the rate. In part D, the process of anaerobic respiration is sometimes referred to as fermentation. Industrial fermenters are used to grow microorganisms. These microorganisms produce penicillin, an antibiotic that is used to treat bacterial infections. Explain how named conditions in an industrial fermenter are controlled. Conditions like temperature, pH, oxygen concentration, carbon dioxide concentration, nutrients, and so on can be controlled in different ways. So for the optimum temperature, we use a cooling jacket to prevent overheating. This ensures that the temperature does not go beyond the optimum because as respiration occurs, due to the microorganisms inside, heat is going to be released. The optimum pH, this is monitored and an alkali or acid is added in order to maintain it. For oxygen, if the conditions are aerobic, oxygen is going to be continuously added, and then a stirrer is going to be used to mix for even distribution of the oxygen that has been added. Also for nutrients, there is addition of nutrients that occurs, and again the stirrer is going to mix the nutrients for even distribution within the solution or within the culture medium. Next, they say some antibiotics are becoming less effective at controlling bacterial infections. They want you to explain how bacteria have evolved so that antibiotics are less effective. Before evolution, there could be a random mutation, and this mutation could give a beneficial or advantageous allele to these bacteria. This allele could be resistance against antibiotics. So the bacteria with this allele are going to survive because they will not be killed by the antibiotics, while the others that do not have the allele are going to die. Those that survive are going to reproduce and they will pass on this advantageous allele to their offsprings. This means down many generations, we will have more offsprings with this advantageous allele, and that means the number or the population is going to increase. And when that happens, the antibiotic resistance is going to increase and the antibiotics will become less effective. So this brings us to the end of question 8. Let's continue to question 9. Question 9. Genetically modified bacteria are used to produce the hormone insulin. Explain how these genetically modified bacteria are produced. Genetic modification in bacteria occurs by first isolating a specific gene using restriction enzymes to cut it out from the DNA, and then we can use the same restriction enzyme to cut the bacterial plasmid. When the stem restriction enzyme is used, it leaves complementary sticky ends so that when we place the plasmid and the gene together, it's easier for them to attach. Then we can use DNA ligase, which is going to help us to insert the genes into the plasmid in order to form the recombinant DNA. The plasmid is going to be inserted into selected bacteria or specific strain of bacteria, which is going to be used for insulin production. In part B, explain the role of insulin in the human body. In the human body, insulin is involved in homeostasis, or I could say in negative feedback. When the blood glucose concentration increases, the pancreas is going to detect and release insulin. The released insulin is going to cause the liver and the muscle cells to take up more glucose from the blood and convert it into glycogen for storage. And then this is going to reduce the blood glucose concentration, returning to normal. In part C, some people are unable to produce insulin. This condition is called diabetes mellitus. People with diabetes mellitus control the condition by using insulin injections, controlling their diet, monitoring how much they exercise. Explain why the insulin is injected rather than taken by mouth. Insulin hormone is a protein, and if it's taken by mouth, it's going to be digested because we have digestive enzymes that break down proteins in our digestive system. These enzymes include pepsin and trypsin. They'll break down the protein into peptides and will no longer have insulin as it should be. Next, they say state why people with diabetes mellitus need to monitor how much they exercise. When people exercise, glucose is going to be used in aerobic respiration. So people with diabetes, if they exercise too much, more glucose is going to be used up and their body might end up losing too much glucose in order to support physical activity. And lastly, they state how people with diabetes mellitus may need to modify their diet compared with people who do not have diabetes mellitus. They need to decrease the glucose consumption, and then maybe they could eat more starch because starch has to be digested, so it's going to release glucose slowly so that too much glucose is not released into their body at a single time. So this brings us to the end of question 9 as well as to the end of this whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.